Okay, so I think we will begin. It's already 3. So I'm Florian LEVET. I'm a researcher in the quantitative imaging of the cell team in Bordeaux, at the Interdisciplinary Institute for Neuroscience. And today I will present some work that we have done in order to do quantification or coordinate-based multidimensional and multicolor single molecule localization microscopy data. So the context is single molecule localization microscopy. So if you are, I will just try to, no, okay. If you have some microtubule dataset like 8 left, and you label it with some fluorophore and you activate with a laser, you will activate all the fluorophore in your data. So everything will throw light, and since one point when it goes through the lens, the objective lens doesn't appear as a point but as a airy pattern because of the diffraction limit of the light, you have this kind of image on left, which is a diffracted limited image where you have a resolution of around 250 nanometer. So in order to break this diffraction limit and do super resolution images, one of the techniques that was developed was single molecule localization microscopy. And here the idea is that you will manage to stochastically only activate a subset of the fluorophore of all the protein, meaning that you will really end up with the image at the middle, where you have a few of the fluorophores that are activated and image at a given time. Because of that, you are able to really precisely segment and localize them, for instance, with a Gaussian fitting. And then you can reconstruct a super resolution image using a pixel size that is below the pixel size of your camera, and you have a better resolution. And here you can see with the microtubules. So obviously, it is quite different from the other kind of microscope because now you have a point cloud. You have the coordinate of your proteins. And how could you use that in order to do a quantification of this data? And what people did first was they went to reconstruct super resolution images. So as I just said before, you are going to define a pixel, an image with a pixel size that is below the pixel size of the camera. So if your pixel size was 160 nanometer, let's say that you use a pixel now of 20 nanometer, you project every one of the localization inside these pixels. So you have the intensity in the pixel is the number of localization in this pixel. And then you can do some quantification like pixel-based workflow in order to do your analysis. But there are some problems here. First, the pixel size may affect the quantification. So obviously the image will be different if you have 20 nanometer as a pixel size or 30 nanometer as a pixel size. The pixel dimension is fixed. So you can oversample, well, you will oversample the spatial region where you will have a lot of pixels that are basically an intensity of 0, and you will undersample the denser one where you can have pixels that will have thousands of localization inside. And then you will need to usually use a combination of different techniques, so a workflow in order to do your quantification, and that may be complex to reproduce and generalize to another biological sample, for instance. And so the idea was, how can we use this spontaneous nature of the single molecule localization microscopy data in order to do the quantification? And something that is quite difficult with single molecule localization microscopy is the experimental parameters, because you have a lot of things that will affect the localization density of your data, meaning the number of localizations that you have in your sample, in your data set. So first you have the fluorophore photophysics. Depending on your fluorophore, the photophysics will be different. So you will have a different number of localization per molecule. You have the labeling density, the labeling efficiency, the acquisition time, the background. So there is a lot of parameters that affect this localization density. And for instance, in our team, we had Anne Béguin at some point that developed a high content screening single molecule localization microscope. And you can see that here, I have 4 different datasets that are normalized with the localization density of the one in the left. And so that's why it's 1. But then the dataset at right is at least almost 5 times more localization than the one on the left. So you don't see it like that because of the representation. There are 5 times more localization in one of the data than the other, and that means that your quantification need to be really robust to the localization density in order that you don't change your parameter for every one of your sample. And so here I will present the work that I have done and the last work that I have done, which is Point Cloud Analyst, and why this software. Because that's more than a decade now that I'm working on developing methods in order to do single molecule, to analyze the single molecule localization microscopy data. For instance, in 2015, I released SRTCLR, which was already a software at this time, but you could only uh, open 2D dataset and you can only add one dataset open at a time. Then in 2019, I released colocalization, which has a method in order to do colocalization analysis. You could do 2D and 3D. You could also open several datasets, but there were no 3D visualization. And at some point in 2020, it begins really a nightmare for me to develop with this legacy software because they were not really well thought at an architecture level for me to really manage to do new development and all these kind of stuffs. So in 2023, I redesigned the application. So all software that I have done, I redo it from scratch in order to have Point Cloud Analyst, which is a new software that we released a couple of years ago, which is a lot better for me to develop and for the user with a better visualization, better interaction, better UI, batch processing. You have plugins, so it's really a better software to use and to develop on. And so PoCA is available on my GitHub, so FLEVET. You have some releases, so the last release is still 2 years old now, so I'm developing it quite daily, but I haven't took the time to do any release. It should arrive in the coming months because there are a lot of developments that have been done that are not available right now in the public version, but still you can get this release that is 2 years old and is working pretty well, pretty robust. And also, you have a website where there is a manual that is directly on the website. So first, I will talk a little about the software itself before going through the different quantifications that are possible with the software. So first, it takes a CSV file. So the localization should be in a CSV file, something that is, for instance, thunderstorm is generating when you are doing the localization process. So you drag and drop a CSV file, and this widget will open, where you will be able to see the header and the different column, a preview of the different column of your localization data. Then you will be able to choose which one of the column you want to load. Obviously, you need to load X and Y in order to be able to have 2D dataset or XYZ in order to have 3D dataset. There is also calibration, meaning that if your localization are not in nanometers, some localization software will maybe put your localization in the pixel coordinate. You can apply a calibration in order to put them back to nanometer. And then this is the software that will be open. And you can see that here you have pretty much the more important interaction things that you can do with the software. Here you have a tab localization that will be mean everything that you can do with the localization are part of this tab. So here you have all the features that you loaded that you can filter with the histogram. You can change very easily the LUT that you apply on the localization in this case. And here you will have some action that you can do on the localization. So you have different visualizations that are possible. So first you could have the point rendering, but directly I use sphere impostor and some shading in order to have a better idea of how the different points are. You can do some heat map. So this is 2D, meaning that you are doing a project. If you are using 3D, this is a 2D projection of the 3D dependent on the viewpoint. You can create objects, so you can have surfaces. You can also do ellipsoid fit in order to have the size of your data. And here, this is a small video, but I will do a demo just after, so we can skip this. Importantly, you can do macro with the software. So every command in the software is defined as a JSON file. So a text file basically that can be recorded in the macro recorder. And then, so here you can see a small process where I'm doing Delaunay triangulation and I'm doing some objects. And then this is recorded in the macro recorder. So then you can pass it to the runner, apply it to a new data, simulation 2, and then the macro was applied to the second data. And here you can see that it was really applied exactly with the same parameters that you did during your macro. And so, as I said, I will do a rapid demo of the interaction for now. So here, this is what you would get if you go to the release part or release page of my GitHub, and you go to PoCA, you go to the release page, and then you will be able to download this zip archive that you just have to unzip, and then you can go here, and you will have the executable. So this is the software when you just open it. You can drag and drop a CSV file or go directly to choose it. Okay. Here you can see that already X, Y, Z, intensity, frame, and sigma V are selected. Here we are already in nanometer, so I'm not applying a calibration. And then it will open the data in 3D. So here this is pure simulation. You will have your localization tab here. You can go to filtering display, and indeed you will be able to really filter your data if you want. Change the features that is used for the LUT display. So this is X, Y, Z, but you could use intensity, for instance. So you can filter, can change the LUT directly by clicking on this button. If you want to do real filtering, so here, the data are still there. So if you are doing a quantification, it will take every localization, even the one that you are not seeing here. So if you want to really filter your data, you can go to the toolbar here, and there is a button that is for duplicate localization. If you click on it, now you have duplicated the filter localization, and you can apply your quantification on the filter data. Things that are pretty convenient when you are using with 3D localization is that it's very easy to do some cropping of the data. So here you have three buttons, X, Y, X, Z, Y, Z. When you click on them, you are directly rotating the dataset to this frame, and then you can use this scissor in order to do some cropping. So you define your zone of cropping, and then you are able to really directly go to some data that is of interest, visualize them, and then go back to the full view and do, for instance, a new cropping very easily. In the MISC tab, you have a few possibilities. You can, for instance, change the background color. You can change the grid that is displayed here, that is giving you some idea of the size of the data set. So you can either use the number definition, so divide by 5 here, because this is 5, but you can also have a length definition. So here that would be, for instance, a line every 100 nanometer. And so it can really help to have an idea of the size of your object, especially after doing some cropping, because now you can really see that you have something that is around 1 to 100 nanometer, basically, for this sphere. And then this is, I think, all that I wanted to show about the interaction. Just the last thing, if you are doing a cropping like that, you can have this view, for instance, and then you can save the camera position, go back to the full view, and load back the camera position, and then you are back to exactly the view that you saved. So it can be pretty convenient in order to go back to some region of your data that is really interesting for you. So I was here, and now I will begin to talk about how you can do really your quantification with point cloud analyst. So usually you would want, when you are using single molecule localization microscopy, you would want to do some either clustering or segmentation when you are quantifying one color. So clustering is really statistically that characterize a small aggregates of molecules that are in your data, usually by comparison with a random distribution of the molecules. So it's really statistically relevant here. You don't want to use a threshold in order to find your object. You really want to have something that is statistically robust comparing to a random distribution. And then there is segmentation, where you will try to classify molecules in different classes with respect to some defined attributes. And this is the case, for instance, for DB scan or tessellation. So you can see clustering as one application of segmentation in the sense that you can use a segmentation technique in order to identify clusters. Still, clusters are statistically robust and relevant, which is not necessarily the case of segmentation. So for clustering, I will present the carry-play function, which is the most used function in the field. So you really try to describe the average number of molecules that exist near another molecule within different radius. So it computes the deviation from a spatially uniform distribution, and this is computed iteratively for varying radius R. So here, for instance, you can see two datasets, one that would be a random distribution, one that would be with some clusters. And you have this equation that defines the carry-play function. So it's pretty easy. It means that for every one of your localization, you will compute the number of neighbors in the vicinity of the radius R. You will divide by the average density. You do the sum of all that, and you divide by the number of localization. So it's really statistically determines the radius of maximum aggregation of the molecules because you know the numbers that you will expect in a given radius if it was a random distribution. So the problem with that, with this carry-play function, is that it's very difficult to understand. So usually what we are using is the hash replay function, which is a normalized version of the carry play, because when you are using the carry play function, the number that is expected is p r square, so it's very difficult to understand. But when you normalize it with this equation, now the number that you would expect for random distribution is 0 with any given radius. So if we have some example here, you have a clustered dataset and a complete random spatial distribution for the second dataset. We go to this small region. We decided, for instance, to compute the carry play function between 50 and 100 nanometer every 10 nanometer. So this is the first one, let's say 50 nanometer. So this is the radius. You compute the number of neighbors inside this radius for all the localization with this function, this equation, and you end up with this result. So for the cluster data, you have a higher value, and for the random distribution, you have something that is around 0. You have now a bigger radius. You still compute your value. You see that the value for the cluster data is higher than before. The one for the random distribution is around 0 again. You do that for a new radius. You have the new point, and you do that again until you have done for all the radius that you have defined. And here you will have this kind of curve. So for a complete random distribution, you have a curve that is really around 0. While for the one that is clustered, you will have a peak and then going back to 0. And sometimes it go even below 0 because there is possibly some repulsion at higher radius. But then, because you have a peak, the peak in your curve will these will define the radius of maximum aggregation that you have in your data. And so it has been used in a lot of different papers. So here is one of the example. And so the problem is that if you have something that is well clustered, like the dataset above, you have a clear peak. Here in this case, it's around 100 nanometer. But when you begin to have different level of organization, and this is the case in the data that are below, because you have some background, you have an adhesion site, and you have cluster inside the adhesion site and in the background. Now you can see that the hash replay is not working very well, and there is no clear peak. So maybe we have some peaks that could be defined here, but this is very difficult to really see. You have no clear peak, so you cannot really use replay, hash replay in this case. So when you are using hash replay function, one of the big advantage is that it's intrinsically normalized, meaning that it's robust to the localization density. So it will give you a statistical robust result of the aggregation in your data, which would be the radius of maximum aggregation. So this is good, for instance, when you want to try to do comparison between different conditions. We'll find if one of the two conditions is more clusters or not, but maybe you will be interested in the distribution of your cluster, and this is not something that the replay or the hash replay function will give you. And then it's sensitive to multiple levels of organization. So as I said, if there is more than two levels of organization, it won't work. So basically, usually people are not using it on the whole dataset, but we will use it on some ROI where there is two levels of organization, basically background and clusters. So even if in the end, even if the method is really very without bias because you don't give any threshold, right? It's statistical or robust. You still have some bias because you will do it on some ROI that you choose. Then we can go to the segmentation technique, and the one that is the most used in the field is DB-scan, so density-based spatial clustering analysis with noise. So it's a segmentation technique. It's suitable for segmenting object and clusters because it's only dependent on the threshold that you will use. It organizes the localization with respect to density in three classes: core points, density reachable points, and outlier points. And I will give some example after. And it uses two parameters, radius R, that is the neighborhood size in order to compute some kind of a density, and a mean points, which is the mean number of points in R to be a core point. So for a point to be classified as a core point, which is different of the mean number of points of a cluster. And with that, you can really do some nice segmentation of things that are not isotropic, as you can see. It's only dependent on the parameters that you will define. So if we have this data and we say that mean point is foo, that includes the current point, if you compute it for this point here, the number of points inside the radius is 5. So 5 is higher than 4. So this point is a core point. If you go to this point, you have 2 points. So it's not a core point, but because of one of the point is a core point, you have a density reachable point. And in this case, the point is alone. It's a outlier point. You do that for all of your localization. You end up with this classification. And usually in single molecule localization microscopy, we're only using two classes. So both core point and density reachable point are designed as object point, and outlier point is designed as background point. So if we go to this small data set here, where you have some cluster, you can define a radius R of 35 nanometer, mean number of point of 15, and you know that the density in this case is 400 molecules per micrometer square, and you end up with this nice segmentation. The problem is that if you go again to this focal adhesions dataset here, the density now is 1,900 molecules per micrometer square, so a lot higher. Because of that, you manage to segment the cluster in the background, but then you segment the whole adhesion site. Obviously, you can change the parameter in order to make shows that you segment the cluster inside the adhesion site, but now you begin to lose some of the cluster in the background. So DB-scan is certainly the most used technique for segmentation in the field. This is because you have two parameters, so you can really fine-tune your parameters in order to manage to do your segmentation. But then two parameters is still annoying because you can change the two parameters, meaning that you have set of parameters that will be different but end up with the same result. So sometimes it can be quite difficult to manage to really fine-tune your two parameters. And the second problem is that it's not normalized. So you need to change the parameter if your density is changing. Maybe not, but in some cases, for instance, if you have 5 times more localization, you may need to change your parameter in order to be sure that you are able to segment all your data. Another strong family that is used in order to do segmentation in single molecule localization microscopy is tessellation. And the first example that was used was the Delaunay triangulation, which is a subdividing space technique that is using triangles constructed from molecule coordinates. So if you have this data, you end up with this triangulation. So basically, you are meshing all your localization, and you are sure that no triangle will overlap with each other because this is the way you construct your Delaunay triangulation. So now it was used in 29 by Badley et al. And in this case, they are defining a global cutting distance. So for instance, in this case, the cutting distance will be 25 nanometer. So it's working very well with the cluster in the background, but again, we'll really segment the adhesion site. And as for DB-scan, you can change this cutting distance in order to manage to segment cluster in the background, but you will lose some cluster in the to manage to segment the cluster in the adhesion site, but you will lose some of the cluster in the background. And so you have exactly, in the end, the same problem as for DB-scan, is that this method is not normalized, so you will need to change you may need to change your threshold if the density is changing, if the localization density is changing. Another problem, at least in our mind, is that the relation between the triangles and the localization is difficult to envision. So how could you use the triangle in order to define and to add relevant features to the localization? This is difficult because one triangle connects three localizations, and one localization is connected to a lot of triangles. And because of that, we decided to use the Voronoi tessellation, which is a dual of the Delaunay triangulation, meaning that one can be reconstructed from the other. So if you have this set of localization, you can compute the Delaunay triangulation. And then the Voronoi tessellation can be constructed from it. So the Voronoi tessellation is also a space subdividing technique. It's also anisotropic by nature because it's only dependent on the affinity of your localization. And what we think is really interesting here is that you have a bijective representation between the localization and the Voronoi cell, meaning that you have one localization, one Voronoi cell per localization. So if you have this Delaunay triangulation, you can construct the Voronoi diagram as you can see here. So basically you are constructing polygons around your localization, and you can see that the edge of your polygon is really the bisectrix of the edge of the triangles of the Delaunay triangulation. So it's not exactly like that the Voronoi diagram is constructed. There is better algorithm to do the construction, but that's the basic idea. And in the end, any point inside the Voronoi cell is closer to the localization from which the Voronoi cell is computed than to any other localization. And so with that, you have two very interesting features that can be computed on the Voronoi diagram. First, you have the connectivity. So you know the direct neighbors of any localization. And what we mean by direct neighbors are neighbors that share one of the edge of their polygon. So in this case, you have 6 direct neighbors for this localization. And then there is the scalability, meaning that the denser the localization are, the smaller the polygon will be. And so you are not, as for reconstructed super resolution images, where you will use a pixel and have a lot of different localization inside the pixel. Here, one polygon equal one localization. And so what we show in 2015 is a way to use all these features in order to compute a local density on your localization. and we generalize this computation. So if you are at 0 rank 0, you are at one localization, the number is 1. You know the area of the polygon, so the density is 1 divided by the area of the polygon, so the area in yellow. But now you can go to the rank 1 because you know the direct neighbors, so the number will be 1 plus 4 neighbors 5. You know the area of this polygon in yellow and gray, and so the density is 5 divided by the area in yellow and gray. And then you can go to the rank 2, compute exactly the same features, and you can go to the rank K. In our case, we are usually using the rank 1 because it kind of smooths a little with respect to the rank 0, and it's easier to segment. And in this case here, I have colored the different polygon with respect to the local density that was computed at rank 1. And one thing that is really interesting with the Voronoi diagram is that you can do a kind of Voronoi normalization. So it's not a mathematical normalization, but it's a normalization that is accepted in the field of people that are using Voronoi diagram. So here you have two simulations, exactly the same, but that would be the same for datasets that have similar organization, spatial organization. And we have a ratio, density ratio of 1 to 5. So the data right is 5 times more localization than the one on the left. If you do the computation of the rank 1 density and you plot the density for all the localization, and here it's a logarithmic distribution of this density, you can see that the frame of the denser dataset is shifted to the right. This is expected. So if you apply absolute threshold, and in this case it's quite easy because you have two peaks in your distribution that make it very clear where you should put your threshold, it will really work well with the localization density of 1, but then the same threshold will work really badly with the denser localization density. And this is something that is expected. But now what you can do with the Voronoi density is that you can normalize the Voronoi density that you have computed by dividing this density by the average density of your dataset, meaning the number of localization of your data divided by the area. When you do that, you can see now that the two frames are shifted exactly to the same frame, right? The two distribution of the density are shifted to the same frame. And now you can apply a threshold of 0, meaning 1 times the average density since we are in a logarithmic distribution, and have a nice segmentation for your two datasets. So this is well for segmentation, but at the same time, there were people in Strasbourg that were using Voronoi in order to go back to clustering, so to go back to statistical determination of the clustering. And to do that, they use Monte Carlo simulation and really simulation of a complete spatially random distribution of the data. So if you have your experimental data, here is simulation, but that would be the same with an experimental data. So you have n localization and you know the area of your data. You can now simulate complete random distribution of the localization, complete random spatial distribution of your localization, and you can do that, for instance, 500 times, and it will give you this kind of data sets. You compute the Voronoi diagram, you use the area of your Voronoi cell, and you plot that. So you have the distribution of the polygon area of your experimental data, and then you have the distribution of all the polygon area for all of your simulation. You plot together the experimental data and the mean of all the simulation. You find where they intersect. This is your threshold. You apply it, and you have a statistical determination of the clustering. You see, the problem with that is that this is very long because they used MATLAB implementation of the Voronoi diagram. So if you need to do it 50 times, 100 times, depending on the number of localization, it can take a lot of time in order to do your analysis. And what I showed a couple of years ago is that this distribution of the spatially random localization can be approximated by a customized gamma function, so in 2D and 3D. And so if you have this data in 3D, you can really use this formula in 3D in order to compute the distribution, so an analytical distribution of your volume of your Voronoi cell, and you can see that it's very similar to the one that you will get through simulation. So now you are able to really apply your experimental data, your analytical function, find where they cross, and do the segmentation of the clusters. And here you can see the same segmentation, but with the simulation, and they are pretty much exactly similar. And so now the interest is that you have no added cost in order to do the clustering as soon as you have done your Voronoi diagram. You don't need to do 100 simulation. You can directly get the clustering from the Voronoi diagram. But still, when you have some data that are deviating from cluster, and this is for instance the case for microtubules here, you can go back to the Voronoi density in order to apply a threshold and manage to do some segmentation. And so if you have this data set again, so that's the adhesion site, you compute the Voronoi diagram, you compute the local density, you apply 1 times the average density, and you end up with this segmentation. But then you will say, okay, you managed to segment the cluster in the background, but then you also segmented the whole adhesion site. Yes, that's true. But now what I explained on the whole data, you can also do it for each of the objects. So for the adhesion site, you can compute the average density of the adhesion site, apply 1 times the average density of this adhesion site in order to have a second level of segmentation and manage to do the clustering inside your adhesion site. And you can have that very robustly because of the Voronoi normalization, fix your different threshold and apply it to the same, to all of your sample on the same condition. And so this we have done, we used this technique in order to do quantification of some synaptic protein, in particular the number of AMPA receptor in nanodomain. We also use that in order to do some automatic segmentation of a range of concentration of antibodies that were acquired with our high content screening, a single molecule localization microscope. We also show that we could use the distribution of the local density in different protein, so pre and postsynaptic, in order to find that they were organized differently. And since 2015, the software was available on my GitHub. And so it was used by dozens of groups in the world in order to do quantification of very different biological models. So this is mostly 2D at that time, but at some point we begin to be interested in trying to do that in 3D also. And so when we started to work on that was around, I think 2016, 2018. There were very few methods that were available in order to do 3D quantification of single molecule localization microscopy. In particular, there were Vivid Storm and 3D cluster vision. Vivid Storm was using DBScan. I think it was DBScan. 3D cluster visu was using Voronoi tessellation, but you can see that these two methods were horribly long, like hours in order to quantify a maximum 100,000 localization. So not really something that you can use in a biological lab. So we really started to try to find better way, faster way to do that. So I started to try to find some libraries developed in C++ in order to do Voronoi in 3D, and I found a couple of them, like Voro++. The problem is that this method were using some grid or k-nearest neighbors in order to speed up the construction of the Voronoi cell, and this is something that is only working with an homogeneous distribution of the localization or the point, while in single molecule localization microscopy, we are in a highly heterogeneous distribution. So we couldn't use this kind of libraries in order to do the Voronoi, the construction of the 3D Voronoi. And so I get back to the fact that the Voronoi diagram is the dual of the Delaunay triangulation, and so that you can construct one from the other. So from a set of localization in 3D, I'm using an algorithm from the library Seagull, which is a very efficient algorithm in order to compute a 3D Delaunay triangulation. It's a multi-threaded algorithm, meaning that you are able to construct the 3D Delaunay of millions of points in dozens of seconds. And then I can use the information of the Delaunay triangulation in order to send everything to the graphic card and create the Voronoi cell on the graphic card. And so this is something that is very efficient and very fast. And you can see, for instance, that even in the million range of localization, we are around 1 minute and 30 seconds for 1 million localization. So very efficient. You can have your Delaunay that is constructed in a couple of minutes for huge data, and then you are able to do your segmentation. And for instance, this is something that is done in real time. So I'm creating the Voronoi diagram for this dataset that is 1,600,000 localization. And basically, this will be done around now, I guess. Yeah, now. So it was around 20, 25 seconds, I think, for this number of localization in this case. And then here you have an example for 4 million localization of microtubule, and it took 1 minute and 12 seconds in order to compute the Voronoi in 3D. And then you can compute the local density, and then you can do your segmentation. So one thing is that in order to do that, I'm using CUDA, which is something that is specific to NVIDIA cards. So you need an NVIDIA card in order to use the 3D Voronoi with PoCA. Still, in PoCA, there is an efficient implementation of DBScan. So here you can use DBScan in 3D, and even with 1,600,000 localization, it was something like 80 seconds in order to do the analysis of this with DBScan. Obviously, with DBScan, there is something. This time will be dependent on the radius that you define. So the bigger radius you will define, the longer the definition, the computation of the DBScan will take. But here you can see some nice segmentation that were done with DBScan on this mitochondria dataset. So I'm going back to some demo. So here we are back to our localization datasets, right? And here you have different buttons. So you have the Delaunay triangulation and the Voronoi diagram. So here we can compute the Delaunay triangulation. So here, this is the Delaunay triangulation in 3D. So you can put the display as fill. So you can see every one of the edges that are connecting all the localization in 3D, right? So you can go back to here. So this is the volume of the triangle, of the tetrahedron, sorry. And in this case, this is again a logarithmic distribution. And so here you can do some selection of your tetrahedron, or you can directly use the cutting distance as I explained before in order to do your selection. Then here you have a few parameters, so the mean number of lock, the mean area, the max number of lock, or the max area of your object. So here I want at least 10 localization in order to be an object. And then I can go to this button here, which is the create object button. And then it will create my object. Okay, so I get rid of the display of the Delaunay. I can get rid of the display of the localization. And here you have your different object. So you have the different distribution of the features that were computed on the object, like the volume of the object, the surface of the object when they are 3D object, the size. So the size is, if you go to one object, for instance, you can see that we are doing a fitting, an ellipse fitting of your different localization. And we use that in order to compute the size as being the mean of the three axes of the ellipsoid. And so you have also the three axes, the major, the minor, and the minor too. And obviously, you also know the number of localization that is inside your object. So you can do Delaunay, and as before, you have you can very easily change the LUT of your object or select the features that you want to use in order to apply the LUT. when you are here, okay, so you have the localization. You can also create a Voronoi. So here, the Voronoi is never shown. If you want to show it, you need to go in the parameter and click on create cells here. So it's not done directly because the process that is used in order to compute the volume is a little different in the GPU. We are not creating exactly the cells. If you want to create them, it's a process that is done on the CPU, so it's a lot longer than creating the cell on the GPU. And here you can see still that you have the density that was computed. We can put it as a logarithmic distribution, and then here you have the density of the localization, and now you can apply a factor, as I said before, in order to do the selection of your localization that you want to keep in order to create the object. You can also use still the cutting distance between under the hood. You know the direct neighbor, so you can compute the distance between the direct neighbor, and then you can use the cutting distance if you want, as for the Delaunay triangulation. But in this case, I just apply the factor, and now I can create new object and have this kind of result. So this is for Delaunay triangulation or the Voronoi diagram. You also have dB scan, as I said before. So you have the distance, which is a radius, the mean number of lock in order to be a core point, and then also a mean number of lock in the cluster. So when you are doing your cluster, if the cluster is below this number, you will not keep it. So if we use this parameter, you can see that we manage to do a nice segmentation of all the cluster here. Okay, you have some features, so the size, the number of detection, major and minor axes. So they are not real objects as for PoCA, like in this object list. And if you want to do that, you can use this button here that will create the object exactly as before. That you could then use the tab object list in order to modify and select and filter as you wish. And finally, so this is 3D. Finally, if you are using 2D, okay, you can also compute the carry play function for this kind of data. So for instance, between 50 and 200 nanometer, every 10 nanometer, and then you can compute the curve. As you can see, this is a carry play here. So this one, you cannot really manage to understand it. But if you go to the H replay, you can see here that you have a maximum at around 60 nanometer in this case. And then you can export your result if you want. Okay, I think I will go back to the presentation. Okay. So after all that, all this work on the segmentation, we begin to be interested in colocalization analysis. So really trying to find correlation between the organization of the protein into color. And so if you are going to conventional fluorescent microscopy, so here you can see two color with the magnification of the same region here. The gold standard for colocalization analysis with fluorescence microscopy are Pearson and Manders coefficient. And this is because they are very easy to understand and very easy to use. And they are based on the correlation analysis or um, ah, I don't find my English term. Comparison between the intensity of the pixel in the two color, because you know, since the image has the same dimension, that one pixel corresponds exactly to the other pixel in the other color. So when you watch this pixel, maybe you can see that it's a high intensity in channel 1 and quite high intensity in channel 2. And if you do that for all of the pixels in your image, you can end up with this kind of scatter plot. And this will give you an idea of the correlation between your two images, meaning that the Pearson coefficient will have a value between -1 and 1. 1 is highly correlated, -1 is exclusion, and 0 is not correlated. Another way is to use Mander's coefficient, which is more a quantifier of the coefficient, while the Pearson correlation coefficient was an indicator. So meaning that the Pearson coefficient is really good when you are comparing different conditions. And in the case of the Manders, you have really a quantification of the colocalization. In this case, you need to apply a threshold for the Manders in order to do some kind of selection of the region of interest in your image. And then if you watch this, so you have two kind of object in the color one object in the color two. If you compute the Manders coefficient for the color one, that would be yellow divided by all the area in magenta, while the Manders coefficient 2 will be yellow divided by the area in green, meaning that in this case, you have a high correlation, a high colocalization for the second color, because this object is highly colocalized to this one, but a low colocalization for the first color because this one is not colocalized at all with any object. So in the case of single molecule localization microscopy, there were already a few methods that were developed in order to do the colocalization analysis. One of the first was the Gettys and Franklin framework, which is very similar in the end to Carrier function. Not exactly the same way to compute the thing, but still it's the same idea. And so you will define a radius. You will compute the value of the Gettys and Franklin in your color, but also in the other color. Then basically L means that you are computed some kind of a density, right? So then you can do the scatter plot of all your value for all of your pixel, for all of your single localization, sorry. And then you can apply a threshold to that in order to really find the region that are highly colocalized. Another method is a coordinate-based colocalization. So in this case, you are using a radius, but now you're also defining a number of beams. You compute the number of localization in each of the channel, so channel 1, channel 2, for each of the points, right? This is a process for one point, but you will do that for all the points. So you find the number in each of your ring, and then you do a Spearman rank correlation, meaning that you will not compare this value here, but the rank when you sort the array. Because if you compare really the value, like that's what would you do if you use a Pearson correlation, that means that you expect a linear relationship between your two colors. And in single molecule localization microscopy, because of the problem of the localization density, the fluorophore photophysics and all that, you cannot really expect a linear relationship between the two channels. So you are using the Spearman rank correlation because you are not comparing the absolute value, but the rank in a sorted array. And in this case, still you will have a value between -1 and 1 as for the Pearson coefficient. And in this case, for instance, we have quite a high correlation, sorry. In the end, you do that for all your localization, you end up with this kind of distribution of the correlation between 0, -1 and 1 for all your localization, and then you can apply a threshold in order to manage to compute the colocalization. The problem is that all this, we have seen that they are not perfectly robust to the localization density. And even in the case of the coordinate-based colocalization, you have several parameters to use. reduce the number of bin and the threshold, so it can be quite difficult to manage to use that. And so what we wanted to do is bring the colocalization gold standard to multicolor single molecule localization microscopy data. That means the Pearson and Menders coefficient. And in order to do that, we decided to use the Voronoi diagram as a proxy for the pixels that you will use when you have Pearson and Menders coefficient. So if you have these two data, you can compute a Voronoi diagram per channel, so per color. So if you are going to this point here that is selected, obviously you know the density in its color because this polygon was originated from this localization, so you have the direct link. So you know that in the first color, this is the identity. But in the end, you know exactly where this point is falling in the Voronoi diagram in the other color. So you can know now that this point has an identity in the channel 1 and a low density in the channel 2. And so we are really able to add a pair density descriptor to the localization. If you do that for all of your localization, and then apply a threshold like for the Menders coefficient by using the robust threshold that I just explained before on the localization density, on the Voronoi density, you end up with this kind of classification where you can see that the red and yellow localization are highly colocalized in both color, so high density in both color, while the cyan and yellow localization are high density in one of the color, and then black is background or not interested, not interesting. And then you can compute the Menders coefficient in the two color, the Menders one being the sum of all the density of the red localization divided by the sum of all the density of the yellow and red localization. And for the Menders 2, the sum of all the density of the blue localization divided by the sum of all the density in the cyan and blue localization. And so what we have shown is that it's very robust to the localization density. So to do that, we use a control that was very simple, let's say. We did a DNA paint acquisition of microtubules that was 10 million localization. We split it, this dataset in two, meaning now that we have exactly the same structure that is labeled with these two datasets. And then the second dataset, we randomly remove some localization from 100% of the localization density to 10%. And here you can see that when we computed the Menders coefficient on this dataset, we had something very, very stable, even when we removed the localization. The second control was showing that when you have a 3D dataset, it's better to do 3D colocalization analysis, because otherwise, if you do 2D, you will have some colocalization that happen, that occur while it's not occurring in real because of the 3D data. And then we applied colocalizer to this data that were provided by Olivier and Greg at the Institute, that they already analyzed in 2014 using reconstructed super resolution images. So they used a pixel-based workflow that was quite complicated, and they managed to find some trend between the different proteins. And in our case, we applied colocalizer with the same threshold, that was 1 times the average density, to all of the conditions, and we had the similar trends than them, but with a method that was a lot easier to use and that can be used also for other kind of biological models. And so you have colocalizer, but you can also do some object colocalization. So you have your data, you do some segmentation, and then you can compute the overlap in 2D or in 3D in order to have the object colocalization happening in your data. Another possibility is that maybe your data are not overlapping, right? So your two proteins may not overlap, so you cannot compute an overlap, or you cannot compute a Menders coefficient. And in this case, you can use nearest neighbor in order to compute the distance between your two proteins. So in this case, we have here already segmented two different proteins. We have a reference protein. We define the distance maximum distance, and then we link all this, all the object of the second color to the first one. So obviously we are giving some distance, so the distance to outline and the distance to the centroid, and we don't link objects that are more than this distance, maximum distance. And so now we can again go on a small demo. So if we are opening again our data, I'm using simulation because it's easier to show. Okay, so now we have two data, two simulation, two color, let's say. If we want to put them together in order to do some colocalization analysis, we have this button here that help to link two datasets in order to do some colocalization analysis. So you can see that you can link four data, but for now the colocalization analysis is only done between the first two. So basically, if you add more, it's more just for visualization. So now both colors are together. And you can see that now on the tab, there is two buttons, 1 and 2, that were not there before. And so you can use them in order to switch between the two colors. So if we go to the localization tab, filtering, I'm on the first color. I will put magenta color, and I will put a white background. Now I can switch to the second color and choose green. Okay, so now it's a lot easier to see the two colors, that there are a little colocalization between these two simulations. So now I can go again to this toolbar and go to the Colloc Tesseller button, click on it. Okay, so now we don't see anything. It's just because the display of Colloc Tesseller is below the display of the localization. So we need to get rid of the display of the localization in order to see the result of Colloc Tesseller. And in this case, there was no threshold applied, so everything is black. But now we have a tab colocalization that is here, and we have Colloc Tesseller that is here. And now we have the scatter plot, so I didn't do exactly the same visualization that on the presentation, where you had the scatter plot that was separated per color. In this case, both scatter plots are together, and I doing a coloring with respect to the density. But this is the scatter plot that I explained before, and then you can apply a threshold. So select directly, and then apply the threshold, and end up with this kind of analysis. And you have the manders also that are computed, and also some Spearman rank correlation. And here you can very easily go to your data in order to see exactly where the colocalization is happening, and if it makes sense. So really, you can then apply as you wish and see how it will change your result or not. Okay. Then you can also do some object colocalization. So I open again the two data. I will do the line triangulation on the two data. I will apply a cutting distance in order to get the object in both cases. Okay. I will do the color object. Go to the object and change the color. And compute the colocalization of the object with this button here on the toolbar. Again, object colocalization. And now in the colocalization tab, I have the object tabs that appear. And basically, that means that every one of these overlap is itself an object. So if I go and I put everything as fill, you can see that in this case, you have the computation of the overlap here. And this is really an object, so you can get all the stat of this overlap. And then I think if I'm going, for instance, to some 2D, oh, yes, this is more dense, okay. But maybe the Voronoi diagram won't work. I think I have a bug right now. Let's try. Oh, yeah, I think I have a bug with the Voronoi in 2D right now. I need to, I know it. I need to put it in the next release. So let's try. With this. And here you can see something that if it's called X pix or Y pix, it's not recognized as X or Y. So you can go to the column here and choose X and choose Y in order to have your data. You can do that the same for the second one. So here I can use Delaunay triangulation again in order to do the segmentation. Okay. I can again do the colocalization object. And here I have what I said before, which is the nearest neighbor test, quantification. So you say which is the reference. You give a maximum distance, and then you can compute and you have your distance, the different distance between your object. So a small thing, PoCA is developed in C++. So it's a compile software. That's why you have the zip and you can unzip and use the exe. But obviously, most of people in the community is using Python. And so PoCA is able to execute some Python interpreter through NumPy arrays. So meaning that you can have a Python script with some quite define how it should be coded. And then you can use PoCA in order to execute a Python interpreter, execute this script with some NumPy array here and get the result and show the result. In this case, for instance, it was in order to get the sphere fit or the ellipsoid fit of all this data. But then I managed, for instance, to the Python interfacing with Camel, which is a machine learning method that was developed by in the team of Dylan Owen. And so here, this is machine learning and CNN. So it used quite big libraries, and I still managed to run it. And here, for instance, you can see that using that was using TensorFlow with the Python interpreter. And I got this bad result, but it's because I didn't do the training of the model. I just use a pre-trained model. And so we are pretty much at the end of my presentation. So I was maybe a little fast, but usually I'm doing it not on remote, but with people. So I guess the demo version is longer. I guess you can maybe, if you want, ask me question about the software, or I can show more some things that you may not have understood. So it's pretty much up to you what you want to do.